This conference will now be recorded.
Good evening, students. I'm Dr. Divya from TeenEinstein. So now we are in the 6th chapter, molecular basis of inheritance. In this chapter, in the previous classes, we were learning about DNA, RNA and all. So we have learned all these things. I'll just give a summary of this. So you know that nucleic acids are long polymers of nucleotides. And you know DNA is the storage of, or that is storing the genetic information, that is the genetic material. And in some cases RNA is also working as a genetic material. So though DNA and RNA both functions of genetic material, but which one is more stable? DNA is more stable because structurally and chemically that one DNA is more stable, that shows more stability. And RNA is the first evolved material and DNA formed from RNA. So first evolved material is RNA and DNA was derived from RNA. Then the double-stranded helical structure, in that in between opposite strands, here you can see the strands, both strands in the DNA structure, right? So these strands, they are held together with the help of hydrogen bonding. So there is a hydrogen bonding between the bases from the opposite strands. The rule, how is the rule? The rule is, or what kind of rule is existing? That adenine is pairing with thymine, A and T, adenine and thymine. Then that is with 2 hydrogen bonds. And next is guanine with cytosine, and that also with 3 hydrogen. Adenine pairs with thymine, that with 2 hydrogen bonds, and guanine with cytosine through 3 hydrogen bonds. So this makes one strand complementary to the other one. So this hydrogen bonding that makes, and the complementary base pairing, that makes one strand complementary to the next one. So the DNA replication, how the DNA is replicating, it is a semi-conservative replication. And this semi-conservative model, that process, it is guided by the complementary base pairing and that hydrogen bonding. So the a segment of DNA that is coding for RNA, that particular one, that coding for RNA, and that forms a protein, and that protein is getting expressed. Okay, so that makes the changes. So that coding region, that is called as a gene. Okay, so let's move to today's class. So after this we have learned in the last class, right? So in today's class, what and all we are going to learn? So after this, we have learned about different aspects of that molecular biology or molecular basis of inheritance. So you know that nucleic acids, nucleic acids are nothing but polymers of nucleotides. So these nucleic acids, DNA, or what is the full form of DNA? Deoxyribonucleic acid, and RNA, ribonucleic acid. So in that DNA is storing the genetic information, but RNA is helping to transfer the information. In DNA, genetic informations are there, but DNA straightly that can't forward that information in the form of a protein. So what DNA does? DNA forms RNA. So RNA helps to transfer this information, and they will get expressed in the form of a protein. We have already told the expression of this gene or a gene, how it is getting expressed in the form of a protein, or RNA, which is coding for a protein. That is the expressive way of a genetic information. So DNA and RNA both are working as a genetic material. Then DNA you can see as a genetic material in most of the organism, almost All the organisms have a DNA as a genetic material and RNA. Where and all you can see RNA in some kind of viruses and all you can see now. It's very familiar like COVID, coronavirus. That is what RNA. In that genetic material is RNA. And DNA and RNA both are working as genetic material. But DNA is the best or better genetic material because it is having stability chemically and structurally. That is having stability or that is more stable. So RNA is the first evolved genetic material and DNA is derived from RNA. So all these things we have told very clearly. Then the double-stranded DNA that is having hydrogen bonding between the bases of opposite strands and the rule, how is the rule? It's like adenine paired with thymine with 2 hydrogen bonds, then guanine with cytosine with 3 hydrogen bonds. So why I am repeating all these things? Because for today's class, we are going to learn about genetic code and mutation and all. So it should be very clear, all these things, the pairing and how it is getting replaced. If it is getting replaced, what and all will happen. So it should be very clear. And we are going to learn about the formation of this codon, triplet codon. So how it is forming an amino acid. So up to that, it should be very clear. Then only you can understand today's portion. So thymine with adenine and thymine with 2 hydrogen bonds, then guanine with cytosine with 3 hydrogen bonds. So that makes the complementary strands. Okay. So the DNA replication, it is a semi-conservative model or semi-conservative way. So we have already discussed about all those things. And the segment of DNA that is coding for RNA, that is called as a gene, right? So then we learned about types of RNA and process of transcription also. So we learned about 3 types of RNA, rRNA, that is ribosomal RNA, that is the most abundant and largest, right? Ribosomal, note down all this because these are important points for your NEET exam. Okay, ribosomal RNA, rRNA or ribosomal RNA, then tRNA, then mRNA. mRNA is the messenger RNA, rRNA is the ribosomal RNA, and tRNA is the transfer RNA. In the rRNA or the ribosomal RNA, that is the most abundant and largest RNA or the largest one is the rRNA. Next is tRNA. tRNA is the transfer RNA and this is the smallest RNA. And next is the mRNA. mRNA is the messenger RNA and this is least abundant. So there are 3 types of RNA: mRNA, messenger RNA, rRNA is the ribosomal RNA, and tRNA. tRNA is the transfer RNA. And abundant and largest form, that is the rRNA, ribosomal RNA. Okay, and least abundant is the messenger RNA. Okay, so in bacteria, there are 3 major types of RNA. Which are they? mRNA, rRNA, and tRNA. mRNA is the messenger RNA, rRNA is the ribosomal RNA, and tRNA is the transfer RNA. Okay. So for protein synthesis, all RNAs are needed. mRNA is needed, rRNA is needed, and tRNA. All these are required for the synthesis of protein. Or when an RNA is getting expressed in the form of a protein, all these RNAs are required. mRNA is required, rRNA is required, and tRNA is also needed. So there is a role for each type of RNA. In the protein synthesis, there is a role for mRNA, rRNA, and tRNA. Got it? Then, in bacterial protein synthesis, this mRNA provides the template, and tRNA brings the amino acid, and the tRNA reads the genetic code. Okay. Then rRNA, that is having structural and catalytic role. When during the translation process, that rRNA or the ribosomal RNA is having structural and catalytic role during the translation process. Okay, so note down all this. In bacterial protein synthesis, all mRNA, tRNA, and rRNA, all type of RNAs having specific role. So what does mRNA is doing there, or mRNA is providing the template for the synthesis. Okay, mRNA providing the template. And tRNA, what tRNA will do? This tRNA brings amino acid and then reads the genetic code. Then last, the rRNA. mRNA role is it is providing template. Then tRNA brings the amino acid and reading that genetic code. Then rRNA is having structural and catalytic role during translation. So in bacteria, transcription of all type of RNA, it is catalyzed by a single DNA dependent RNA polymerase. So catalytic role, it is catalyzed by a single DNA dependent RNA polymerase. Here you can see that RNA polymerase. So that is DNA dependent RNA polymerase. Note down, in bacteria, transcription of all type of RNA, that is catalyzed by DNA dependent RNA polymerase. Very important for NEET exam, DNA dependent RNA polymerase. That is the, in bacteria, the transcription of all these type of RNA, it is catalyzed by single DNA dependent RNA polymerase. In the case of bacteria, I am saying the transcription, it is catalyzed by single DNA dependent RNA polymerase. Then what is happening there? By binding to that promoter, here you can see, to that promoter, that RNA polymerase that initiates the transcription. Okay, it is binding to the promoter region. So that RNA polymerase that initiates the transcription process, and that step is called as initiation. So what is initiation? That DNA dependent RNA polymerase that is binding to the promoter region. Promoter, from that term itself, it's very clear. Promoter, it is promoting that process or translation process. Okay, so DNA dependent RNA polymerase that is binding to that particular region or the promoter region, and that is called as initiation. So that step is called as initiation in transcription. We are talking about the transcription. Okay, so here in this stage also, that is following the rule of complementarity, complementary base pairing. That rule is followed everywhere. In this stage also, that is following the complementary base pairing rule. So that here this uses nucleoside triphosphate as a substrate. So for an enzyme, here DNA dependent RNA polymerase, that is an enzyme. An enzyme reacts with the substrate, right? Enzyme substrate complex will form, and then finally the protein formation, all these enzymatic reactions. So how it is taking place? That uses nucleoside triphosphate as a substrate. Nucleoside triphosphate. Note down this. This is not for your state board exam. It's for, especially this is for these points for your NEET exam. From this you will get a lot of questions. So that uses nucleoside triphosphate as a substrate. So which is the substrate here? Nucleoside triphosphate. And how it is polymerizing, or how that polymerizes? The polymerization is taking place in the template dependent fashion. Okay, here you can say the template dependent fashion. This part, the DNA dependent RNA polymerase, it is binding to the promoter region, and the substrate, which is the substrate here, that uses nucleoside triphosphate as a substrate. And how it is polymerizing? That polymerization takes place in a template dependent fashion. It is depending on a template, and in that template depending fashion, the polymerization is taking place. Then next step comes as elongation. Next step is called as elongation. Here this is initiation, that transcription process that is getting initiated in the first stage. Here it is getting initiated. And next is elongation. See, now in initiation it is getting attached to a point. Now that should get elongated. That should move, right? So the next step comes elongation. So while continuing to the next step, elongation, that somehow that facilitate to open that helix. Here you can see. Here that makes that helix to open. Helical structure is getting open here. So while continuing to the next step, elongation step, what you can see, the double helical structure is getting open, or it is opening that helix, right? So still, you can see a short stretch of RNA still remains bound to that enzyme, right? Here you can see that RNA, a short stretch of RNA, it is still remaining there, which is bound to that enzyme. So it is getting, or a RNA is remaining there only. So that nascent RNA falls off, and when it is falling off, once the polymerization reaches the end, then what happens when that polymerization reaches the end, or the termination region is reaching here? When it is reaching near the end or the termination region, all these are happening. So nascent DNA falls off and the RNA polymerase also falls, right? So here that nascent, here you can see that RNA still remains there. So that and all, when it is moving towards the termination region or the end of the process, this RNA, that nascent RNA, that will fall off, or that will go, that will move away. Okay. See, here you can see. In the first step, initiation step, that RNA polymerase, here it is getting attached and it is promoting the process of transcription. Then that is the initiation step. Next, elongation, this helical structure is getting open and that RNA, that is still remaining there. So when it is moving towards the termination, or at the end of that reaction, this is the termination step, you can see that RNA, it is falling off. So here in the third picture, here third one, you can see the termination of the transcription occurs. So do you have any idea about how that RNA polymerase catalyzes all the three steps? Initiation, elongation and termination. Do you have any idea about that? The process of this elongation, it is catalyzed only by RNA polymerase enzyme. Which enzyme? RNA polymerase enzyme is catalyzing the elongation. Okay, the process of elongation is catalyzed only by RNA polymerase enzyme. So to initiate the transcription, the RNA polymerase, here you can see RNA polymerase, that associates with initiation factor. Here you can see initiation factor. So RNA polymerase, to initiate the transcription process, that RNA polymerase, the enzyme, DNA dependent RNA polymerase, that attaches, or that is getting attached with the initiation factor. Which is that initiation factor? Sigma factor. Here you can see the sigma factor. So the RNA polymerase is getting attached to the sigma factor for the initiation process. Okay. So to terminate the transcription, next to terminate the transcription, here the termination process, RNA polymerase associates with the Rho factor. This RNA polymerase associates with the Rho factor, and that is the termination factor. So which is the initiation factor? Sigma factor, that is called as the initiation factor, and Rho factor is the termination factor. So to initiate, RNA polymerase is getting attached with the sigma factor or the initiation factor, and to terminate, for the termination of this transcription, this RNA polymerase is getting attached with the Rho factor or the termination factor. So to either initiate or terminate, the association with the sigma factor or the Rho factor, that makes, or that determines the specificity, or that alters the specificity of RNA polymerase. Okay, the sigma and Rho factor, that becomes functionless during elongation. Here, sigma factor or rho factor, both won't work. During which step? During elongation, both don't have any role. They are functionless in the elongation process. Okay. So in bacteria, we were talking about the bacteria transcription process. So what and all we said? Initiation, elongation, and termination. So during initiation, first step is initiation, then elongation, and last one is termination. Here, DNA-dependent RNA polymerase, that is binding to the promoter region, and to which factor it will bind? To the sigma factor. That is called as the initiation factor. Sigma factor is the initiation factor. Then, next step, it is moving to elongation. Elongation means when it is attaching to a point, initiation point or the promoter region, it is getting attached, and from that process is getting that elongates, right? That time the sigma factor don't have any role in that. And that RNA, it is moving away. Okay, so when it is reaching that DNA dependent RNA polymerase or that transcription process, it is reaching near termination or at the end. See, now from here that reached near the end. So at the end of this process, this DNA dependent RNA polymerase, it will bind with the rho factor. What is the rho factor? That rho factor is the termination factor. So RNA polymerase getting attached with the rho factor, that is the termination factor, or that helps to terminate the transcription process. So that time this RNA, this nascent RNA, that will fall off or that will get separated. Okay. And in the termination step, this RNA polymerase also will get separated or that will move away. Okay. So there are 3 steps: initiation, elongation, and termination. And all these steps are catalyzed only by RNA polymerase enzyme. Okay. In the case of bacteria, there is only one enzyme, RNA polymerase enzyme. So sigma and rho factor, that becomes functionless in which step? During elongation step, this sigma and rho both are becoming functionless. In the case of elongation step, sigma factor and rho factor, both the initiation factor and termination factor, both will become functionless. So that's all about the process of transcription in bacteria. And next, so in bacteria, only one RNA polymerase you can see, right? So in the transcription process of eukaryotes, there are some other complexities. You can see some more complexities in the case of eukaryotic transcriptional process. So in eukaryotes, in addition to this RNA polymerase that found in the organelles, okay, there are at least 3 RNA polymerases in the nucleus. So there are some extra RNA polymerases in the nucleus in the case of eukaryotes, but in the case of bacteria, there is only one RNA polymerase, okay, that functions throughout. And here in the case of eukaryotes, there you can see clear division of labor. Okay, in the case of eukaryotes, there are at least 3 RNA polymerases in the case of eukaryotes. So in the nucleus, at least 3 RNA polymerases will be present in the case of eukaryotes. So there you can see a clear division of labor. So each work is specifically given. Okay, each RNA polymerase that is having specific function. Got it? Each RNA polymerase, it is having specific function, and specific function is given to each and every RNA polymerase. So how that division is working or division of work is done, that's what I'm going to explain now. And this is very important for NEET exam. So you know that ribosome is having two different subunits. You can see here the picture of ribosome. Here in this diagram, you can see large subunit and small subunit. See the two green color subunits are there here. So there is a large subunit and a small subunit for ribosome, right? So the small subunit. Here both subunits, there are two different subunits. You can see here two subunits and both subunits are required for translation. For translation, both subunits, large subunit and small subunit, that both are having their own function. So the small subunit, what the small subunit will do, that small subunit decodes the genetic messages. Here the small subunit decodes the genetic messages. And the large subunit, what that large subunit is doing? Large subunit that catalyzes the peptide bond formation. Okay, small subunit decodes the genetic message and large subunit catalyzes the peptide bond formation. Peptide bond formation is catalyzed by the large subunit and small subunit will do the decoding of genetic messages. Got it? So the large subunit that catalyzes the peptide bond formation. So in ribosome, we used to say two subunits like 16S, 30S, etc. What is this S? S stands for 16S subunit, we will tell 30S subunit. What is that S? S stands for sedimentation coefficient. This is not there in your book. So just for general information, I'm saying this S stands for sedimentation coefficient. Okay. So what do you mean by the sedimentation coefficient? Sedimentation coefficient is the downward velocity of a macromolecule in the centrifugation. While centrifugation or centrifuging this thing, that macromolecule will get sedimented. So that downward velocity of macromolecule in the centrifugation process. So that higher value, the higher the value, the greater the molecule. If the value is higher, the molecule will be greater. Okay, that's how we can say that. So what is S? S stands for sedimentation coefficient. See here you can see 16S, 30S. S stands for sedimentation coefficient. Okay. So coming back to our point, we were saying about ribosome with two subunits. So in eukaryotes, there is a clear division of labor. Okay, RNA polymerase. We were saying about RNA polymerase. So RNA polymerase 1. What RNA polymerase 1 will do? RNA polymerase 1 transcribes ribosomal RNA. Okay, ribosomal RNAs. Here you can see 28S and 18S and 5.8S. Okay, so ribosomal RNA polymerase 1 transcribes ribosomal RNAs like 28S, 18S and 5.8S. Okay, 5.8S ribosomal subunit. So the ribosomal RNA polymerase 1 transcribes rRNAs 28S ribosomal subunit, 18S ribosomal subunit and 5.8S ribosomal subunit. Got it? Then RNA polymerase 2. RNA polymerase 2 that transcribes precursor of mRNA. The heterogeneous nuclear RNA or hnRNA. So RNA polymerase 2, what that is doing? That RNA polymerase 2 transcribes precursor of mRNA. Precursor of mRNA. The heterogeneous nuclear RNA. Precursor of mRNA. What is that? Heterogeneous nuclear RNA or hnRNA. Heterogeneous nuclear RNA. Note down heterogeneous nuclear RNA or hnRNA. The precursor of mRNA. Okay. Then RNA polymerase 3. RNA polymerase 3 transcribes tRNA 5s ribosomal subunit, 5s ribosomal RNA, and SN RNA. So RNA polymerase 3 transcribes tRNA, 5s ribosomal RNA, and snRNA. What is snRNA? Small nuclear RNA. Small nuclear RNA, snRNA. Okay. So there are at least 3 RNA polymerases in nucleus. So in addition to the RNA polymerase found in the organelles, so RNA polymerase found in the organelles, that is there. In addition to that, there are at least 3 RNA polymerases in nucleus. So there is clear division of labor among these RNA polymerase, different RNA polymerase. RNA polymerase 1 transcribes rRNA 28S, 18S, and 5.8S. Then RNA polymerase 2, that is responsible for transcription of tRNA, 5S RNA, 5S rRNA. Okay, 5S rRNA and snRNA. snRNA. What do you mean by snRNA? Small nuclear RNA. Okay. Then RNA polymerase 2. What this RNA polymerase 2 is doing? That transcribes precursor of mRNA. What is that precursor of mRNA? The heterogeneous nuclear RNA or hnRNA. That is the precursor of mRNA. Okay. Next is so just now we discussed about complexities. Complexities of eukaryotes. In eukaryotes, what are all the complexities? So there is, in bacteria, there is only one type of RNA. But in the case of eukaryote, the first thing is there are different types of RNA. Okay. And there is clear division of labor. And next point is the primary transcription or hnRNA contains both exons and introns. Okay. But they are non-functional. Primary transcripts or the hnRNA that contain both exons and introns. Exons and introns and all we have already discussed. So it is subjected to splicing. So in splicing, introns are removed and exons are joined in a particular order. Right? Exons are retained and introns are removed. Introns are the non-coding region. So if you are removing also, nothing will happen. So introns are removed and exons they will join in a particular order. Okay. So the primary transcript or hnRNA that contain both exons and introns, but they are non-functional. So this is subjected to splicing. So in splicing, what they will do, the introns are removed. Introns are the non-coding region and that is getting removed and exons are joined in a particular order. Okay. So in prokaryotes, splicing is not required. Why? Because hnRNA is absent in prokaryotes. Okay, note down, in prokaryotes, hnRNA is absent, so splicing is not required. Only in the case of eukaryotes, I am saying all these complexities. So, this is hnRNA. Then that hnRNA undergoes another process that is called as capping and tailing. Splicing over. Next process is capping and tailing. What do you mean by capping? Capping means addition of unusual nucleotide to the 5' end. Addition of unusual nucleotide to the 5' end. 5' end of hnRNA, heterogeneous nuclear RNA. Okay, to the 5' end of heterogeneous nuclear RNA. So which nucleotide it is adding? Methane guanosine triphosphate is added to the 5' end of the hnRNA. Methane guanosine triphosphate, that is added to the 5' end of the hnRNA. Then what is tailing? Next, now capping over. What is tailing? Tailing is adenylate residues. Okay, adenylate residues, it is added to the 3' end in a template dependent manner. In a template dependent manner, it is depending upon the template. So in a template dependent manner, the adenylate residues are added to the 3' end. Okay, so like this, hnRNA is fully processed, and after that, it is called as mRNA. hnRNA is the precursor of mRNA, right? So in a productive form, you want to make that hnRNA. So whatever they will do, first splicing, then capping and tailing. Okay. So the second complexity, that the primary transcript contains both the exons and the introns are non-functional. The primary transcript that will be having exons and introns. You can see here the primary transcript that will be having exon and intron. Exon is the coding region, intron is the non-coding region. So if you are removing the non-coding region or the introns, that exons will get expressed, right? So it is subjected to the process called splicing. In splicing, what they are doing, the introns are getting removed and exons are getting joined, okay, in a particular order. So here you can see, in this you can see introns and exons, right? This yellow color region, they are the exons, the coding region, and the red color region, that is the intron, the non-coding region. So without removing these introns, we cannot join these exons. So splicing means the process, the procedure of removing these introns. Got it? So through splicing, the introns are getting removed and exons are joining. Then next step, this hnRNA undergoes additional processing, that is called as capping and tailing. So in capping, what they will do, an unusual nucleotide, which is that methyl guanosine triphosphate. Methyl guanosine triphosphate, that is added to the 5' end. Methyl guanosine triphosphate, it is added to the 5' end. Here you can see 5' end, that is added. Then in tailing, adenylate residue 200 to 300, it is added at the 3' end. Okay, adenylate residue that is added to the 3' end. Here you can see at the 3' end. That is in a template independent manner. In a template independent manner, this is getting added. So it is the fully processed hnRNA. And now we can call this hnRNA as the mRNA, and this mRNA is transported out of the nucleus. So that kind of fully processed hnRNA, we can call that as mRNA, because that can work as a messenger RNA. Okay, that is a processed hnRNA and that can work as a messenger RNA, and that is transported out of the nucleus for what? For translation. Okay. Then, so nowadays the studies at molecular level, that is in a well advanced form, the molecular level study. So it is very easy to understand the significance of this kind of complex matters. So we can understand very easily the significance of all these steps. So the split gene arrangement, it is the ancient feature of the genome. Okay, split gene arrangement. What is that split gene arrangement? Here you can see exons and introns and in between all this. So that is the ancient feature of the genome. So you know that in splicing, the introns are removed and the exons are joined in a defined order. So introns, it is the portion of gene which is getting transcribed. Introns, but here while transcription, introns are getting transcribed. The non-coding region also getting transcribed. But while the process of translation is the protein synthesis, right? So during translation or the protein synthesis, these introns are removed. These introns are removed because introns are waste, or that is the non-coding region. So the presence of introns, that is a reminiscence of antiquity. That is the reminiscence of antiquity. So what is that? Splicing means introns are removed and exons are joined in a defined order. So introns means that is the portion of the gene, and while transcribing this will be there, introns will be there. But while the process of translation, that introns will be removed and the exons will be joined, because introns are the non-coding region. So the presence of introns, that is the reminiscent of antiquity. Okay. So the process of splicing that represents the dominance of RNA world, but the presence of introns, that is the reminiscent of antiquity. Okay. Process of splicing represents the dominance of RNA world, but the presence of introns, that is the reminiscence of antiquity. So from this we can understand that the idea about RNA and RNA dependent process in the living system, that is very important. That is having more importance nowadays. So many research workers are studying, or they are behind these studies. Studies are going on. So we can expect more advanced result from this area. Okay. Next. So now you have almost an idea about the transcription and the replication and all. Next we are going to learn about genetic code. So in the process of replication and transcription, what is happening? And in both cases, nucleic acid is getting copied from another nucleic acid, right? Nucleic acid is getting copied from another nucleic acid. Here you can see how the next strand is getting formed. It is getting, there is a copying process, right? So on what basis it is getting copied? You know that it is forming another nucleic acid on the basis of complementary base pairing, right? So we have learned about complementary base pairing. So on that basis, this is getting copied. So we are getting an idea about the formation of another nucleic acid on the basis of complementary base pairing, right? So this is for the replication and transcription. So what about translation, or what is the translation? You know that translation is the formation of protein from the nucleic acid. From the nucleic acid, proteins are getting formed. That is called as translation. So translation means the transfer of genetic information from the polymer of nucleotide. And that's how it forms a polymer of amino acid. Polymer, monomer, and a monomer means single unit. Polymer means multiple units, right? So translation means transfer of a genetic information from a polymer of nucleotide. The genetic information is getting transferred from the polymer of nucleotide. That means more than one unit is there. From that information is getting passed. And that's how it is forming a polymer of amino acids. Okay. So here there is no complementarity. Okay. There is no complementarity between the amino acid and the formed protein. In the case of translation. In transcription, you can see a complementary base pairing and a complementary strand also. But in the case of translation or the protein synthesis, there is no complementarity between the amino acid and the formed protein. The newly formed protein don't have any complementarity from one particular genetic code or that information, or from that RNA, one protein is forming. There is no complementarity between this. Got it? So if there is any change or any difference in the nucleic acid or that particular genetic material, that may result in the change of amino acid also, or that protein formed, that newly formed protein, that also will get affected, or that also shows that particular change. So that change, this is leading to the preposition of genetic code. That led to the preposition of the genetic code. Okay. How that particular genetic code can code for a particular sequence of amino acid during the synthesis of certain protein. So how that led to the proposition of a genetic code? The particular genetic code can code for the particular sequence of amino acid during the synthesis of certain protein. Okay, so now you got an idea what is the genetic code. Okay, so during replication and transcription, a nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. On the basis of complementarity, replication and transcription, we can say. But in the process of translation, that requires transfer of genetic information. In the translation process, that is the transfer of genetic information from a polymer of nucleotide. From a polymer of nucleotide, the genetic information is passed and it is synthesizing polymer of amino acid. Okay, so there is no complementarity is existing between nucleotide and amino acid, and there is no theoretical basis also for that complementarity. Okay, so there existed ample evidences. Okay, so the change, if there is a particular change in the nucleic acid or a particular amino acid or a genetic material, if there is a change in the genetic material, that will be responsible for the change in amino acid, and that protein, that expressed protein also will change. That also will get changed, and this led to the proposition of a genetic code that could direct the sequence of amino acid during protein synthesis. Okay. Next, that biochemical nature and the structure of DNA, and this and all you have an idea, the biochemical nature of DNA and the structure of DNA, and we had discussed all this about in previous classes, and the starting classes we were discussing about the structure of DNA. Then preposition and deciphering, or deciphering means the decoding of that genetic code. It is not very easy and it's very challenging, right? It's very difficult. So that needs the help of scientists, geneticists, physicists, biochemists, organic chemists. With help of all these scientists, we can decipher or decode the genetic information. Okay, so a physicist, George Gamow, he said there are only 4 bases. George Gamow, a physicist, he argued that there are only 4 bases and they have to code for 20 amino acids. Got it? So these bases, that should be, all these bases, that should be in a particular combination to form different genetic codes. There are only 4 bases, he said, and that should code for 20 amino acids. And how it will code for 20 amino acids? Through different combinations that can code for different amino acids. So according to him, to code for all 20 amino acids, the code should be made up of 3 nucleotides. So how it will code? According to that scientist, George Gamow, the code to code for the 20 amino acid. How it will code for the 20 amino acid? That code should be made up of 3 nucleotides. So this was a major or very, very important proposition made by physicist George Gamow. Okay? To code for that 20 amino acid, that code should be made up of 3 nucleotides. George Gamow proposed this. Okay? So the permutation and combination of these bases or these amino acids, that would generate or that may form 64 codons. Okay, that may form 64 codons. So how many codons? 64 codons. Got it? So who proposed? George Gamow proposed that 20 amino acids and only 4 bases are coding for that. In how it is coding for this 20 amino acids? Through different combinations. So the permutation and combination of these different bases, 4 into 4 into 4, 4 raised to 3. That would generate 64 different codons. And that generates many more codons that required. Okay. Then now we discussed that that scientist, he gave an idea about codon and amino acid, right? But it was very difficult to prove that codon is triplet. How to prove that codon is triplet? So providing a proof for that a codon is a triplet, it was very difficult. So scientist Crick, another scientist Crick, he provided the experimental evidence supporting the concept of triplet genetic codon. So who provided first experimental proof? Scientist Crick. He provided the experimental evidence for supporting the concept of triplet codon, triplet genetic codon. Okay. And the chemical method which is developed by Hargobind Khorana. It was the first instrumental synthesis of RNA molecule. Hargobind Khorana, he developed the chemical method. Okay, and that RNA molecule, with defined combinations of bases. That is Hargobind Khorana's work. Note down all the scientist name Crick. He gave the concept of triplet codon, or he gave the experimental proof. Then chemical method was developed by Hargobind Khorana. Then Marshall Nirenberg. His cell-free system for protein synthesis, that is helped in the deciphering of genetic code. Cell-free system for protein synthesis, that is helped to code this genetic code deciphering, that helped in the deciphering of genetic code. And another scientist, Severo Osho, he discovered polynucleotide phosphorylase enzyme. Polynucleotide phosphorylase enzyme, and that was helpful in polymerizing RNA with defined sequence in a template independent manner. Okay, this discovery was helpful in RNA polymerization with a defined sequence. And how was it? It was in a template independent manner. So enzymatic synthesis of RNA. Severo Osho, he discovered enzyme polynucleotide phosphorylase. Note down enzyme polynucleotide phosphorylase, and this was helpful in polymerizing RNA sequence, and that was in a template independent manner. Okay. So that means RNA is enzymatically synthesizing and RNA is not forming from a template strand. Right, that is the meaning of that. That means RNA is enzymatically synthesizing in this, and RNA is not forming from a template strand. Got it? And here, next you can see the table of codons for various amino acids. Okay, you know that codons are triplets. Triplet codons. So first letter from this line. Here from the left side line, and second letter, this is the second letter in the codon formation. First letter you can select from this, and second letter from here, and third position letters are coming from the right side line. Okay, so from this checkerboard you can write easily the combinations of genetic codons. The different genetic codon combinations you can write from this. So for example, from left side you are selecting U, this one, as the first letter. So the first position you can write U, and second position you can select letter U from here. This is the first position U you are writing here, and the second position letter also you are writing from this top line, and you are writing the second position letter U from the top line. Then for the third position you are selecting here also these four bases only. So the first letter you are selecting, the first combination, that is triple U, and this codes for phenylalanine. So that codes for triple U, that codes for phenylalanine. And next here you can see first letter. Again, we are choosing the first letter U. And second letter U. Next, third letter from here. The second one. You are writing C. That also codes for phenyl alanine. Both codes for phenyl alanine. Okay. That's why we are mentioning as PHE. In the second table, it's very clearly written which codes for which amino acid. So here you can make out which one is coding for which amino acid with the codon. The codon is also mentioned here. So you can get a clear idea about first base, second base, and this is the third base. So in that combination, in a particular combination, which enzyme it is forming? From the second table, you are getting a clear idea about that. Okay. So next AUG. Here you can see A from the first line and U from here, second one, and G from the last. That codes for methionine. AUG codes for methionine. Okay. So these are the 64 codons and the amino acids of each codon. So this 4, 4, 4. That combination in different permutation and combination, you can see 64 codons and the amino acids of each codon. Okay. Next you can see the features of this amino acid. Features of the genetic code. So you know that codons are triplet, right? So the codons are triplet, and among these 61 codons. These are point wise I am explaining. So there is nothing more to study. So exact points you have to note down. It will be useful for both board exam as well as NEET exam. So codons are triplets. So among these codons, 61, we said totally different permutations and combinations that make 64 codons. So out of that, 61 codons coding for amino acids. And 3 codons, they are not coding for any amino acid. And these are functioning as stop codons because they are not coding for any amino acid. So when that codon comes, that works as a stop codon or nonsense codon or a termination codon. That is called as a stop codon or nonsense codon or termination codon. So which are those termination codons? UAA, UAG and UGA. UAA, UAG and UGA. These are the termination codons. So every codon that is unambiguous and specific. So what do you mean by unambiguous? Means one codon, one amino acid. That is, there is a specificity. One codon and one amino acid. There won't be more result from one codon. Okay. That means one codon codes for one amino acid. And the code is degenerate or the degenerate or redundant. That means some amino acids are coded by more than one codon. So by more than one codon, some amino acids are coded. Here you can see serine, proline. So different codons are coding the same amino acid, right? Leucine, here UAA, UUA, that code for leucine. Even UUG, that is also coding for leucine. Even phenylalanine, UUU, triple U and UUC, both codes for phenylalanine, right? So that's why we are saying more than some amino acids are coded by more than one codon. So this is called as degeneracy or redundancy. So for 20 amino acids, 61 codons are coding. So in a contagious fashion, that means next or together as a sequence, that codon is read in mRNA. Okay, and there is no punctuation or there is no separating dot or comma between the sequences. Okay, so that is together, that is working as a sequence, and that codon is read as an mRNA. Together it is working as an mRNA, or that is read as a sequence. and there is no separation or there is no dot or no comma between this sequence. And the next point is that code, this genetic code, that is almost universal. Genetic code is almost universal. So for example, either in bacteria or in human, whatever may be, the codon UUU, triple U, that codes for phenylalanine. Okay, phenylalanine, that codes for phenylalanine, either in bacteria or in human being, whatever may be, it codes for phenylalanine. Okay, so these are some exceptions. Some exceptions we can see in the case of mitochondrial codon and in some protozoans. Some exceptional cases are there. Biology means some exceptional cases will be there. So, some exceptions have been found in mitochondrial codons and in some protozoans also. In human mitochondria, AUA codes for methionine. For AUG, that is having dual function, that is acting as an initiator codon, and that codes for methionine also. So, in human mitochondria, AUA codes for methionine. For AUG, that is having a particular, or that is acting as an initiator codon, and that codes for methionine. Okay. And next topic is mutation and genetic code. Mutation studies. You know that mutation studies that help to understand the relationship between gene and DNA. So through mutation, when mutation comes, then only you can understand what is the relation between a gene and a DNA. So what is mutation? Some kind of changes. So studies by making some kind of changes. Some kind of changes where we need to make in gene. Then only we can make out the changes which is coming in the codon, and finally in the protein also that change will come. So finally what kind of changes that makes one organism, that we can see. If there is a change in the codon, that will come in the produced amino acid, and finally that will be expressed in the form of in the particular protein which is formed. That is also getting changed due to that particular change in the or mutation. So due to that protein, expressed protein also will show some kind of difference. So that protein expression, difference in that protein expression, that will make some kind of changes in that particular organism also, right? So imagine in a segment of DNA, large deletion or addition is happening. In a DNA segment, there is an addition or deletion, something is happening. The effect of that addition or deletion, that is very easy to understand. How we can understand from this example here, this example I am going to explain you. So addition of a gene. That may lead to the loss or addition of a gene. Thereby, the function of that particular gene also will get changed, right? So point mutation. Now we are talking about point mutation. What is the point mutation? A mutation in a genetic mutation in a particular point. That means a single nucleotide or a single base is getting changed. It is getting inserted, it is getting added, or it is getting deleted from the DNA or RNA sequence. That kind of pointed change, that is called as point mutation. So this kind of mutation or a point mutation that affects only one or a very few nucleotides. There is only in one point it is getting changed. So that affects only a one or few nucleotide in that gene sequence, right? So such a change, that type of a change in a single base pair. So in a gene, a single base pair is getting changed, but that is resulting in the change of that amino acid, right? If a single base pair changing also, amino acid may change. We have learned 64 different codons, and that codes for different amino acids also. Hope you have heard about sickle cell anemia. So in sickle cell anemia, in the change of a single base pair in that gene, that is for beta globin chain. in beta globin chain, and that change results in the change of amino acid. So in sickle cell anemia, a single base is getting changed. That also in the beta globin chain, it is getting changed. So that change in the beta globin chain is resulting in the change of amino acid. So that instead of glutamate, which amino acid first it should form? Normal case, it should form glutamate. So that change or mutation in that single base, that changes the product to valine. Instead of glutamate, valine is getting produced. So this results in the diseased condition called sickle cell anemia. Instead of glutamate, valine is getting produced. That is the reason for sickle cell anemia. Okay. So just for an example, I'm explaining here. Here you can see the statement RAM, Ram has red cap. Now there is some meaning for that sentence. All these are having three letters: RAM, Ram, HAS, has, RED, red, CAP, cap. So consider these three letters as a codon. This is Ram, it is a codon, RAM. HAS, has, that is another codon. RED, red, that is another codon. CAP, cap, that is another codon. When it comes together, this is having a particular meaning, right? So in a codon, here, in a codon, mutation happens. Like addition or deletion, it is getting added, some base pair is getting added or it is getting deleted. So in this case, we are inserting a letter. Imagine we are inserting a letter to this one. Okay. Then after rearrangement, how we can read? We are inserting a letter B, another letter B between has and red. So after this, RAM is correct. Then has, after has, this HAS, that particular codon, we are inserting a letter B. So here, in between has and red, this HAS and RED, in between that, we are inserting a letter B. So how we can read? RAM, that is okay, as such. And has, has is okay. Then B, next comes the inserted B. So triplet codon, how we can, if we are inserting B, here RAM, here this is as such, next HAS also same, but inserted B is coming in between this, right? So B, instead of red, RED, next triplet codon, how it will come? BRE. Then here one B is getting inserted, so BRE. Next DCA and T. Here you can see DCA, this is moving right side. DCA, this is becoming next codon, part of next codon. And T, it is separated. So here insertion, that makes changes in the next codon series also. So the whole sentence is becoming something different, right? Now it is becoming meaningless, or there is some kind of changes. So it won't give the exact product. So that's what happening in the case of sickle cell anemia. Instead of that, some other product is getting formed. Okay. So that mutation, that makes changes in the genetic code, and finally in the product that makes, or the protein formation, expression of protein, that expressed protein is also getting changed. There is some kind of changes in the protein expression, and finally that affects the organism in the form of this kind of diseases or something. Okay. So from this exercise, from that previous exercise just now I explained that we can understand that when we are inserting or deleting one or two bases, only we are deleting, then also that whole reading frame, it is getting changed. If you are inserting or deleting whatever, maybe the whole reading frame is getting changed. So such type of mutation, they are called as frame shift mutation. Frame shift insertion or deletion. Frame shift mutation, it is of two types. That may be insertion or deletion. Insertion mutation is also there, deletion mutation is also there. Because why it is called as frame shift? It is shifting the entire reading frame, right? The entire reading frame is getting changed. So this is a proof that codon is triplet. The triplet codon form, if there, something is getting added, that entire reading frame is getting changed. So how we can say this? How a reading frame is getting changed? By the insertion or deletion of one or two bases. Then where this deletion or addition is taking place? by 3 bases or the multiple of 3. From there onwards, the reading frame remains unaltered. If there is a 3 bases are getting added or deleted, or there is a multiple of 3, then there won't be much changes in the reading frame, or the reading frame remaining unaltered, or there won't be much change. Exactly 3 bases are adding. There is an additional codon. If it is getting deleted, then also one complete codon is getting deleted. Then it is not getting, complete frame is not getting altered. Only that particular portion is getting altered, right? So, is it clear? So, triplet codon. This is the proof for triplet codon. So, mutation that makes the complete idea about the triplet codon. Okay. Is it clear? So, we have got a clear idea about gene, genetic code, mutation, and how it is getting, or how it is giving a proof for the triplet form of a codon. Got it? So, that's all about today's class. Thank you.